Welcome. Today, we are diving into a really revolutionary idea for fighting some of the world's toughest diseases. We're talking about targeting proteins that scientists once thought were completely untouchable. The real challenge, though, is how do you drug something that's practically invisible? Let's get right into it. It sounds like a riddle, doesn't it? But honestly, this is the core problem facing drug developers right now. You see, the targets they need to hit aren't these nice, solid, stable things. No, they're much more like fleeting ghosts or shifting clouds inside our cells. So, how in the world do you design a drug for something that barely even exists? These proteins have a nickname, undruggable, and here's why. Traditional drugs, they work like a key in a lock. They need a very specific, well-defined pocket to bind to. But the proteins causing some of our worst diseases, they don't have a stable lock. They're shapeless, kind of ambiguous, which has made them pretty much impossible to target. Well, until now. To solve this incredible puzzle, scientists had to look way deeper into the secret life of proteins. And it turns out the key wasn't in the final shape of the protein, but in the amazing journey it takes to get there. When we see proteins in a textbook, they're always shown as these beautiful, static 3D sculptures. But that's kind of like looking at a finished piece of origami and totally ignoring the whole folding process. The journey is every bit as important as the destination. So let's start at the end of that journey. This final functional shape is called the native state. This is the protein in its active form, ready to do its job in the cell, whether that's being an enzyme or providing some kind of structural support. But how does it get there? Well, a protein starts out as this long, floppy chain of amino acids. It doesn't just snap into its final shape. Nope. Instead, it follows what's called a folding pathway, a really complex sequence of twists and turns to find its most stable form. And all along that pathway, the protein takes on these temporary, fleeting shapes. These are the folding intermediates. These are the ghosts we were talking about. They're these transient structures that exist for just a split second before the protein continues on its journey to its final native state. And this visualizes that journey perfectly. You can actually see the protein chain going through all these different shapes. And it's not a random process. It's a guided series of steps. Every single one of those shapes you see before the final active state. That's one of those fleeting ghosts, a folding intermediate. And as we're about to see, it's also a potential target. This whole process, this complex, delicate dance, is a biological marvel. But it's also a point of critical vulnerability. What happens when just one step in this intricate dance goes wrong? It turns out you don't need some huge error to cause a total catastrophe. A single change in the genetic code, just one tiny point mutation, can completely alter the folding pathway and stop the protein from ever reaching its correct final shape. Let's look at a really specific example here, the A1AT serpent protein. Okay, so for this thing to fold correctly, a crucial salt bridge has to form super early in the process. This bond, the one highlighted between a glutamate and a lysine residue, it acts like an internal magnet, guiding the rest of the protein into its proper structure. And this just lays out the problem brilliantly. In the healthy, normal protein, you have an attractive force. The glue and lys pull together. Simple. But in the Z mutant, that glutamate gets replaced with another lysine. So now you have two positive charges that are actually pushing each other apart. That one little change from attraction to repulsion just derails the entire folding process. The protein misfolds, and instead of doing its job, it starts clumping up with other misfolded proteins. These clumps are toxic. They build up inside cells, gumming up the works and leading to really severe liver and lung disease. So if the problem starts way back in the folding pathway, maybe the solution does too. This single insight has led to a completely new way of thinking about drug discovery. Instead of fighting the final monster, what if we could stop it from ever being built in the first place? This quote from the research just lays out the whole strategy. Instead of trying to block the final, undruggable protein, what if you could intercept it during its folding process? What if you could trap it in one of those fleeting, intermediate ghost states? This new approach is called PPI-FIT. It stands for Pharmacological Protein Inactivation by Folding Intermediate Targeting. As you can see in the diagram, the whole goal is to design a tiny molecule that recognizes and latches onto a pocket that only exists on a temporary folding intermediate, not on the final protein. Okay, so here's how it works, step by step. First, the protein starts to fold. Then, our special little drug molecule binds to it. This one action traps the protein in that intermediate state, blocking it from finishing its journey. And here's the really elegant part. The cell's own quality control system spots this trapped, misfolded protein and flags it for destruction. So you're not just blocking the protein, you're actually tricking the cell into getting rid of it completely. The implications for this are just massive. By learning how to drug the folding pathway itself, we are opening up an entirely new frontier for medicine. I mean, this is so much bigger than just a single new pill. It's a fundamental shift in strategy. For decades, countless proteins have just been written off as undruggable. This new playbook means we can go back and look at all of those old targets again, but this time with a completely new set of eyes. Just think about what this could really mean. We're talking about potential new treatments for diseases caused by these misfolding proteins. That includes things that are currently untreatable, like prion diseases, cancer targets that have resisted every drug thrown at them, and a whole host of neurodegenerative disorders. By targeting the journey, not the destination, we may finally have a way to fight back. The potential is just staggering. This entire breakthrough came from looking at a process we thought we understood, protein folding, in a completely new light. By learning to see the ghosts in the machine, we've actually found a way to drug them. It really makes you wonder, right? What other invisible targets are hiding in plain sight, just waiting for us to shift our perspective?